researcher at Yale School of Medicine. I also have an affiliation with the Amsterdam University Medical Center. So before we start, I have two disclosures. One is that I'm a co-founder of a company called Boundless Bio, and that company is aiming to design drugs against cells containing ecDNA. You'll hear a lot about that later on. So that is a conflict of interest. All right, so my lab studies brain tumors, in particular glioblastomas, but also lower grade gliomas. We think of glioblastomas as grade 4 or high grade gliomas. Now, the somatic alterations that drive these cells, that drive this disease, have been extensively mapped, amongst others by an effort called the Cancer Genome Atlas, the TCGA. So I now have to explain what TCGA means, because the audience increasingly consists of scientists that have started their careers after TCGA ended. So I now tend to explain that it stands for the Cancer Genome Atlas. So I'll get back to the TCGA a little bit later, actually, but suffice to say that TCGA basically sequenced up to 500 patients with a glioblastoma. And from that work, they or we, because I was part of that team, we identified that basically all glioblastomas have somatic alterations in 3 key pathways. First, there is the receptor tyrosine kinase pathway. Oops. Then there's the p53 signaling pathway, and then finally the RB1 or cell cycle pathway over here. And we first reported this in 2008 in this very first Nature paper from TCGA, and then we confirmed this, but in a more elaborate fashion in a follow-up paper a few years later. So what you might notice if you review the pathways and you review the genes that make up the pathways and the way in which they are being altered, you might find that in many cases, those genes are being altered through gene amplification. EGFR is the most common example there. Nearly half of glioblastomas contain a focal amplification of EGFR. But there are numerous other genes, including PTGFR8, MET, CDK4, CDK6, and a few others. So glioblastoma, by and large, is a disease that is driven, at least in substantial part, through gene amplification. Now, gene amplifications can happen in multiple ways. For example, through linear duplication, as is shown here, we have an amplification of a region that makes the chromosome a little bit longer. And because of that, it might show up in cytogenetic stains as being a homogeneous staining region, or an HSR. So an HSR from a cytogenetic stain is essentially a focal amplification, the replication, the duplication of a particular gene sequence numerous times, leading to this homogeneous staining. Now, another way through which oncogenes may be amplified is extrachromosomally. In this case, a chromosomal region gets spun off the linear chromosome and somehow self-ligates. And because of its self-ligation, it now has formed a circle. And this circle contains an oncogenic sequence. Now, in this instance, it's not that the sequence is repeated on the same structure. It is that you get multiple circles, and each circle contains one copy of the oncogene. So now you have achieved an amplification of an oncogene, but through a circular extrachromosomal DNA. And in older literature, you might find that those extrachromosomal DNAs were called double minutes. So double minutes is basically another term for extrachromosomal DNA. Nowadays, we often shorten extrachromosomal DNA and just say ecDNA. So I'm going to use ecDNA for the rest of this talk. Now, a brief overview of what we know about ecDNA in cancer is that these are circular DNA elements that range in size from approximately, these are not absolute numbers, but approximately 50 kilobases to about 5 megabases. A very key characteristic of ecDNA, and I'll have more data to support this later, is that they do not contain centromeres. And this has a big impact on the behavior of ecDNAs. And then we should never forget that ecDNA is first and foremost a mechanism for amplification of oncogenes and oncogenic elements. So how did we get here? How did my lab ended up being studying ecDNA? What was our motivation to ultimately end up there? So to get that, I think I need to explain a few things about my trajectory as a scientist. So I studied several decades ago, sort of before formal coursework on bioinformatics and computational biology existed. I studied biomedical sciences, or my major was biomedical sciences. And because I had a lot of friends doing computer science, I also did a major in informatics. So basically pretty hardcore computer science stuff. And because of the combined background, I was increasingly interested in studying informatics, but in a biological context. In other words, bioinformatics. But as you might notice, I've put biomedical sciences first because that was my very first interest. And I still, to this day, focus on biology first. And for me, data analysis is just a tool to help understand the biology. And that's exactly how I did my undergrad training. I first studied biomedical sciences and then took on computer science / informatics coursework because I thought that would be a good way to help me understand the biology of the diseases that I was studying. And I should also say I was pretty terrible at the bench. So then I moved on to do a PhD, and in the PhD, I studied expression profiling of leukemia. So I was fortunate to end up in a lab that had established a massive collection of leukemia samples and had performed expression profiling of all those leukemia samples. So all I needed to do as the only bioinformatician in that entire institution, in that entire hospital, is take the data set, do some fancy cluster analysis, etc. And basically that was my PhD. It was actually a fairly straightforward PhD. And it only took me 3 years because of that. Now, as part of those 3 years, I spent 3 months as sort of a visiting scientist in the Quackenbush lab, the John Quackenbush lab at the Dana-Farber Cancer Institute in Boston. There's actually a funny little story there in that sometimes these funny little stories help determine the rest of your life. So Quackenbush, who's still a professor at the Farber, or at least at Harvard Medical School today, at the time was in Baltimore at what is called the Tiger Institute. I don't think it exists anymore, but it used to be the Tiger Institute, a famous institute for gene mapping and gene sequencing. So he was there. I was interested in working with him. So I wrote him an old school letter via snail mail and sent it to him to see if he would accept me. And time went by and I didn't hear anything back from him. So I wondered, is he not interested or did he just somehow miss the letter? So then I actually sent him an email and he responded within 5 minutes, like, you sent me a letter? What were you thinking? Which I thought was funny. But then it turned out that he's actually moving his lab from Baltimore to Boston. And that's how I actually first ended up in Boston. And I've often wondered if Quackenbush had not moved to Boston, if I had tried to find a postdoc in Baltimore and how different my life would have been. So Quackenbush did move to Boston. So when I did my visiting research internship, I spent 3 months at Dana-Farber. And because of that, I started to interview for postdoc positions in the Boston area, also recognizing that the Broad Institute was truly a phenomenal environment for a budding computational biologist like myself. And I ended up starting my postdoc with Matthew Meyerson in 2007. And in fact, that was April 1st, 2007, was the day that my postdoc at the Broad Institute started. Now, I already hinted at the TCGA. So the TCGA, the Cancer Genome Atlas, was this ambitious project funded by NCI as well as NHGRI to perform initially gene sequencing of cancer. And the idea was we've figured out the human genome. Now, by comparing cancer sequences to the reference genome, we're going to now figure out exactly how cancer works, and we're going to cure all cancer. And this is why combined the two institutions. I thought I could get a mouse here somewhere. Maybe this. The two institutions decided to, NCI and NHGRI, which are two NIH institutions, decided to launch a $100 million grant opportunity for mapping of the cancer genome. And as you might see, The actual project itself, so initially the discussions, basically the preparation phase started in 2005, but actually the funding was announced in 2006 in November, and which means that the actual grant started another few months later. And as a matter of fact, it started on April 1st, 2007, which is by coincidence the exact day that I started my postdoc. And Matthew's lab was one of the, I think it was 6 labs or 6 teams that was funded to work towards this mapping of cancer sequences project. So only 6 places had been funded, and Matthew's lab happened to be one of them. And the project started the day I started my postdoc, which was rather fortuitous. Because the very first disease, I skipped a slide here. So the 3 first diseases that NIH or NCI proposed to tackle with the Cancer Genome Atlas was, in chronological order, the brain tumors, ovarian cancers, and the lung cancers. So the day I started, we had just received a first batch of brain tumor specimens, and those were making their way through the analyses pipelines, at which time there was expression arrays and copy number arrays, and then also some Sanger-based exon sequencing. So about 15 months after that, the TCGA published its first paper, which was this paper in Nature, Comprehensive Genomic Characterization. This was one of the original comprehensive genomic characterization papers, and you might have seen at least 50 more papers from the TCGA after that with the exact 3 same words in the title, because this paper basically laid out the framework for most subsequent papers. Another little tidbit there is that this manuscript was actually submitted, I remember it well, in late June of 2008, and only because we had learned that the Johns Hopkins Institution, which is in Baltimore, had a paper that was nearly accepted, also focusing on exon sequencing of human glioblastomas. In other words, there was a very prominent competitor. So we hustled, we threw together a manuscript, it was submitted late June, and 6 weeks later it was published, and that's still to my day today the shortest timeline from submission to publication that I'll ever experience. So glioblastoma was the first tumor type in TCGA. TCGA coincided with my own personal start date at the Broad Institute. So it was natural that I started to work on glioblastoma, and in particular, given my background in expression profiling of leukemia, I was a natural fit to work on expression profiling of glioblastoma next. So that's what I did. And long story short, my paper is considered my postdoc paper as a first author is considered the second TCGA paper. Now, it doesn't maybe seem long from September 2008 to January 2010. It's about 16 months. But I can tell you that the cancer and the cancer genomics community was very much excited about the TCGA and was basically very eagerly awaiting for new TCGA publications. And I just happened to be on the lucky end of that. So when my paper got published, there was a huge interest in the topic. And as a matter of fact, my first author paper is actually cited more highly than the original Nature paper. In fact, it's the most cited paper in Cancer Cell of all time. Now, is that because we did brilliant science? Well, probably good science. I mean, I'm not going to sell my own work short. But was it such good science that it has to be the most highly cited paper in Cancer Cell? I'll leave that to others to judge. But obviously, I just happened to be at the right time, and I happened to have the right skill set to be there in that position. So I think that's a sort of something to keep in mind, that you have to be, in order to get lucky, you have to expose yourself to being lucky. And I think that's what happened here. So because of the success of this subtypes paper, I was able to go on the job market, apply for positions to start my own lab, and as a consequence, started my own lab, which was a dry research only lab at MD Anderson Cancer Center. Now, you may ask, how did I go from Boston to Houston? It was because I was keen to stay within the TCGA network, and MD Anderson was also a prominent partner of the TCGA. So this move allowed me to continue to be involved in TCGA. So starting a computational lab in 2010, probably a little different from these days, because 2010 we were still in the middle of figuring out what to do with sequencing data. During my time at the Broad Institute, because the Broad was so much at the forefront of developing sequencing technology, I had been fortunate to be working very closely with the people that were, for example, writing the GATK, or that were writing Picard and other pipelines that we still tend to use to this day. So I had a decent understanding of sequencing data analysis. Nowadays, my grad students can do whole genome sequencing data analysis without too much training, which blows my mind how quickly these methods have been taken up by the community. So at this time, the TCGA was probably the largest and most relevant source of data for a young cancer computational biology lab to work on. This explains, and again, the reason why I wanted to really stay within the TCGA network, so I would have had early access to these data sets, and also to be able to work directly with the people creating the data, because I always feel that if you know how the data was generated, you'll be able to analyze it better. So I joined Bioinformatics and Computational Biology department in MD Anderson and started working on these early TCGA data sets. And these efforts, I was fortunate to hire a few very good postdocs who worked with me in analyzing these data. And one of the first projects we embarked on was to precisely map breakpoints in glioblastoma tumors. You could say it was an early version of calling structural variations or defining the breakpoints of structural variations. And this led to the very first publication of my own lab, which was in 2013, shown here. So in this paper, we focused on breakpoints, and because we focused on breakpoints, we ended up also recognizing focal gene amplifications. And one thing we noticed was these patterns of focal amplification. If you look at this top sample here, you see there's a bunch of adjacent segments that are highly amplified. Here's another bunch of adjacent segments that are highly amplified. But this is not a very clean amplification like the one that's here. Like this is pretty much one segment amplified and another maybe 2 or 3 segments amplified. This is very dirty. This is like many segments that are broken up by other segments and then have been stitched back together. Here's another example, even better, many spread out segments that together form one amplification. So we observed this pattern and also found that, at least for this particular set of samples, these patterns centered around 2 oncogenes, namely CDK4, a well-known cell cycle regulator, and MDM2, a gene that had been recognized as potentially regulating P53. Now, CDK4 and MDM2, a particular MDM2, when we found those genes, we started to learn more about them, because I always believe in trying to understand biology, and realized that MDM2 stands for murine double minute 2. So if you go back to this paper from 2013, and recognizing the term double minute, recognizing the very segmented pattern of amplification, even though we did not have the data to quote-unquote prove that these were extrachromosomal double minutes, we speculated that most of these focal amplifications were in fact double minutes. So this was our very first entry into the ecDNA world. Just around that same time, I had started a collaboration with Anna, who is a research scientist at Henry Ford. She's now an associate professor there. And her mentor, Tom Mickelson, who is a neuro-oncologist. And we started a project where Anna and Tom would collect tumor specimens from patients with a glioblastoma. Anna would then culture those as neurospheres, and she would subsequently transplant the xenograft, the neurosphere cells, into mice in triplicate. And she would then collect specimens from each of these steps, so the patient sample, the neurosphere, and the 3 xenografts, and sent them to us down in Houston, and we would perform sequencing and subsequent data analysis. And the purpose of this project was fairly plain. It was to map the level of genomic drift that occurs when you take patient samples, you culture them, and you put them into mice, to understand how that might impact any kind of drug response studies down the line. So we were basically modeling glioma evolution in vitro and then in vivo. So Anna was in Henry Ford's hospital, and Hoon was the postdoc in my lab working on this project. So here's a landscape figure of this project. You see each of the patients labeled at the top, and each patient basically has 5 columns, namely the tumor sample, the xenograft, the neurosphere culture as the second column, and then the 3 xenografts as columns 3, 4, and 5. And here we indicated the presence and absence of various focal alterations. And you might appreciate that by and large, there's not many changes. For example, a P10 mutation found in the parental tumor here was also discovered in the model systems derived from that tumor sample. And the same is true for most alterations. CDK4 here found in all 5 samples, here as well. CDKN2A found here in all 5 samples, and there's a few other, and here's another example of that. But there are also differences. And one difference that stood out to us was this focal amplification of the gene MET, M-E-T. What we noticed in particular is that this MET amplification appears to be found in the parental tumor sample here, and for this other tumor sample here. Then MET appears to return to a wild type setting in the neurosphere culture in both cases. And in both cases, the 3 xenograft samples subsequently see a return of the focal MET amplification. So here's a zoomed in version of that same pattern. So here we have the 2 tumor samples at the top, the xenografts, the neurospheres at the second row, and then the 3 xenografts for both patient samples. So we have a focal amplification of MET here. It seems to be a very clean 1-segment amplification. The breakpoints, by the way, between the 2 samples are slightly different. So we know that this is actually, these are truly 2 different amplifications. But you can also see that the breakpoints are the same between the parental tumor sample and the 3 xenografts. In other words, there's a clonal relationship between this focal amplification in the parental tumor and the xenografts amplifications. These are not newly derived amplifications. They originate from the original tumor. So based on this, we have to conclude that there's a strong clonal selection process going on here. Namely, in the parental tumor, there's a focal amplification of MET that when those cells are put into culture, are selected against. Apparently, the presence of the MET amplification provides some kind of disadvantage. But a few of those cells may survive, and when you put these cells back into the mouse, in a microenvironment, perhaps they're the ligands for the MET amplification are present again, so maybe that's why. Either way, the cells containing the MET amplification have an advantage now over other cells. So the dominant clone in the xenografts in all 3 cases and all 6 cases is the MET amplified clone. So clonal selection twice, first going into culture, then getting out of culture. Now, we had lots of data on these cases, including exome sequencing data. So we called point mutations. And because of the very strong clonal selection pattern, we expected to see point mutations that would be aligning with the pattern of the MET amplification. We were expecting to find mutations that would disappear and reappear because they would be present in the same clone, in the same cells as the MET amplification cells. But as you can see, we don't really found that. We don't really see mutations that disappear and reappear. So we felt that there was an evolutionary discordance between the way the focal amplification was inheriting versus the way that the point mutations are inheriting. Now, this was challenging. How could we explain, this discordant evolution? And then we had a eureka moment. In fact, the eureka moment happened while we were on vacation in May. So I guess if you want a eureka moment, I encourage you to take vacations in May. In part because we had been working on a breakpoint story, in part because we had been reading lots of literature, we recognized that the very likely explanation for the discordant evolution that we were seeing is that one, the MET amplification happened on an extrachromosomal DNA, and two, the mutations are largely on linear chromosomes. And because extrachromosomal DNA inherits differently, and I'll get back to that, differently from linear DNA, now we see discordance in their evolutionary patterns. So we did fish analysis. We stained cells derived from these xenografts with probes to illuminate the specific MET gene. And as you can see, the MET stain, the MET signal is all derived from areas outside the blue chromosomes, very clearly demonstrating that indeed MET is focally amplified, because you see many green signals, so many copies of MET. And you see that MET is extrachromosomally amplified. So here's the model. We know extrachromosomal DNA elements lack centromeres. Chromosomal alterations reside on linear chromosomes, which of course have centromeres. So during mitosis, the chromosomes are pulled apart and put in and end up in two different nuclei in an even fashion. There's an even split of chromosomes. Now, for extrachromosomal DNA lacking a centromere, there's no place for the centromere to bind, which means that inheritance of extrachromosomal DNA is random, which means that ecDNA gets replicated. So you go from 3 copies of ecDNA to 6 copies of ecDNA. And subsequently, those 6 copies can end up in the 2 offspring cells entirely unevenly, with 6 here and 0 here, all the way to perfectly even. But it's a random process, so each of these has a pretty much an even likelihood of occurring. So this is what I mean by the discordant inheritance. And it also means that if you have cells that are provided a competitive advantage, as we've seen for the cells containing the MET amplification, that one, you get selection for those cells, but two, in this scenario, every time there's a cell cycle, the cells with the highest number of copies will have the highest proliferative advantage. And therefore, those are the cells that end up dominating the tumor in the scenario where there is a proliferative advantage. So you can get very rapid selection of the cells with a high number of copies. And this explains how we could have this very rapid transition from a mostly MET positive to a mostly MET negative to a mostly MET positive clone. So again, this is the same general model. In our situation, we had a patient tumor where the different colors here mark different tumor subclones, but tumor subclones as marked by point mutations. And within each of the subclones, there are cells with and without the extrachromosomal DNA. When we cultured those cells, apparently there was a disadvantage for cells containing the amplification. So the mutational subclones continue to exist. Maybe there's new ones on the basis of acquired new mutations. But the ecDNA cells are selected against. And then subsequently, when put back into the mouse, there's perhaps a new mutational subclone, but the dominant pattern here is that the number of cells containing the ecDNA has taken over the tumor, leaving almost no cells lacking the ecDNA. And if you want to understand tumor evolution, of course, it would be critical to understand these types of processes, because if we would just base our conclusions on the presence or absence of mutations, we would have entirely missed what is actually driving this tumor, which is the ecDNA. Now, because this is a computationally focused group, I want to spend a little time on some larger and perhaps ambitious computational analysis that we have performed. So previous literature had suggested that ecDNA in cancer is relatively rare. Now, our collaborator Vineet Bhavna at UCSD had written a tool called Amplicon Architect that could analyze whole genome sequencing data to detect circular extrachromosomal amplicons. So with Amplicon Architect, and I'll skip the methods, the precise definition of the method, but with Amplicon Architect, you can analyze whole genome sequencing data and figure out which samples contain ecDNA and at which locus. So we worked together with Vineet and started analyzing additional whole genome sequencing data sets. The first step in that process is to identify all regions in the whole genome that have all segments that consist of more than 4.5 copies and that are at least 10 kilobase in size. And then subsequently, Amplicon Architect helps to classify all those amplicons. First, by identifying every amplicon that carries the signal of a breakage fusion bridge, which can be circular, but it doesn't have to be circular. And the signal resembles a circular DNA signal. So in order to be conservative, we first classify everything that carries a BFD signature as a BFD. From the remaining amplicons, those where we can complete a circular path, we'll label those as circular amplicons. And the rest we call heavily rearranged or linear, or of course, there's also tumors like leukemias that do not have any amplifications at all. And then we use the amplification, we use the classification of the amplicons to classify tumor samples. And here we do it slightly differently, namely, everything that has a circular amplicon will be labeled as a circular tumor, then BFD, and then non-ecDNA classes. And what we, in part, were interested in is not just to evaluate the presence of ecDNA in newly diagnosed tumors, but also to understand, which is something we could readily do by analyzing whole genome sequencing data sets from the TCGA and also from ECOG. But additionally, we wanted to understand the role of ecDNA in tumor metastases. I should mention that we had previously mapped ecDNAs across newly diagnosed tumors and found that 14% of newly diagnosed tumors contain one or more ecDNAs. But now we are also interested in understanding ecDNA over a longer period of time. And an opportunity to do so is afforded to us by the Hardwick Medical Foundation, which has performed whole genome sequencing on thousands of advanced post-treatment cancers. And they kindly make all those whole genome sequencing data sets available to us for reanalysis. By the way, this is of course not just privy to our lab. This is a resource like the TCGA. You can apply for access. It's truly a phenomenal resource, and I certainly encourage anybody interested in this to pursue data access. So Hoon Kim, who previously was a postdoc in the lab and who now is an associate professor in Sungkyun Kwon University in Seoul in South Korea, worked together with his student Soyeon to together reanalyze all TCGA ECOG samples and data, and to analyze all the whole genomes we could get access to from Hardwick Medical Foundation. And in totality, this led us to analyze over 8,000 whole genomes, as well as a good number of non-tumor whole genomes for comparison. And the purpose of this exercise was to compare oncogenic amplifications between newly diagnosed tumors and post-treated advanced cancers. Now here we mapped, here we used the classification strategy I explained earlier to identify the relative presence of ecDNA across newly diagnosed tumors. And as you can see, the tumor type that my lab is mostly working on, glioblastoma, is the tumor type that has the highest relative presence of ecDNA. So perhaps it's not so surprising that we were relatively early in recognizing the role of ecDNA in cancer. Had we still worked on leukemias, the odds of us finding ecDNA to be important would have been pretty small. perhaps my lab would be a clonal hematopoiesis lab. Other cancer types where we find a highly frequent presence of ecDNA include ovarian cancer, lung squamous, and esophageal cancer, but also breast cancer has a decent proportion of ecDNA. Now we turn to Hardwick Medical Foundation and find that overall, it appears that there's a pretty significant enrichment in the presence of ecDNA in advanced cancers relative to newly diagnosed tumors. You can actually, if I flip them back and forth, you can sort of see more red in one versus the other. Now, in part, of course, Hardwick has a distribution of tumor types that's different from the tumor distribution in the TCJ Peacock cohort. So we also did a matched comparison. At the cohort level, we found that ecDNA can be detected in this larger cohort of tumors in 16.6%, but across Hardwick, which, as I said, these are advanced, mostly metastatic tumors, the frequency is 26%. So a pretty significant, approximately 62% increase in the frequency of ecDNA. But more importantly, because we have differences in tumor type distribution, if we do a matched comparison, we find that there's 8 tumor types that have a significant enrichment in the relative frequency of ecDNA. In other words, there's a higher likelihood that a metastatic tumor has an ecDNA than that the newly diagnosed tumor has an ecDNA. And this nominates ecDNA as one of the drivers, at least, of the metastatic tumor progression process. And these tumor types include renal cancer, bladder cancer, lung adenoma, colorectal cancer, prostate cancer, esophageal cancer, breast cancer, and finally, cervical cancer. However, we also found a few tumor types where the frequency appears to be significantly decreased, and that is in the sarcomas, the melanomas, and the endometrial cancers. I should, however, flag that if you, for example, think about sarcoma, sarcoma is known to be, you can break sarcomas apart into many subtypes of sarcoma, which tend to be genomically quite different. So for that reason, the frequency of sarcoma is like the decrease of sarcomas, is likely explained by the difference in subtype distribution in the Hardwick versus the TCGA cohort. And this appears to be less so the case for, let's say, bladder cancer or cervical cancer. Now, the genes targeted by extrachromosomal DNA, which are shown here on the left, this is BFB and this is linear, are by and large the same between the primary tumor set and the advanced cancer set. So there's no gene that appears to be frequently amplified in metastasis versus in at diagnosis. Overall, the genes that you see being amplified on ecDNA are the typical genes that you would find on amplifications, which includes CDK4, MDM2, EGFR, cyclin E1, et cetera, et cetera. As we did in our 2020 publication, we evaluated whether the presence of ecDNA would associate with any kind of survival pattern. And when we take the whole cohort, we do not see any particular survival difference amongst primary tumors based on the presence of ecDNA, at least relative to the non-ecDNA tumors. Tumors that don't have any amplicons fare a bit better than tumors containing at least one type of amplicon. Intriguingly, when we limit this analysis to patients younger than 60, we find that the presence of ecDNA actually very significantly associates with poor outcome. And this is independent of tumor type. We did a multivariate analysis to demonstrate that this is tumor type independent. So amongst younger patients, the presence of ecDNA appears to make a prognostic difference. What we thought was interesting is because of the depth and breadth of our two cohorts, the TCJ Peacock primary tumor cohort and the HMF cohort, which we can subsequently further split into HMF tumors that have been treated and tumors that have not been treated, is that we find that treatment appears to associate with the presence of ecDNA. So both for chemotherapy, so these are systemic chemotherapies, the cisplatins, the alkylating agents, the topoisomerase inhibitors, all kinds of those kinds of agents. Pre-treatment with such an agent results in tumors that have a significantly higher frequency of ecDNA compared to tumors that have not been treated. And here you can appreciate the similarity in ecDNA presence between TCGA and HMF tumors that are not treated. And the same is true for radiation therapy. So radiation therapy appears to associate with the increased frequency of ecDNA. And when you now take this group of HMF-treated tumors and you can further split them up into different types of treatment, we find that those tumors that have been treated with multiple different therapy types have the highest relative proportion of ecDNA, whereas the targeted therapies associate with the lowest frequency of ecDNA, as well as the immunotherapies. So therapy appears to be able to drive, and perhaps treatment resistance appears to be able to drive the formation, or at least the detection of ecDNA. And I just want to include this slide. This is the age distribution of the two cohorts, the primary tumors from TCGA PCOG and HMF, to show you that there's no absolute gain in ecDNA as patients are older. So there's no significant difference, there's no significant trend that the frequency of ecDNA increases with age. The point of showing you this is that the association between treatment being able to create ecDNAs, or at least drive the formation of ecDNAs, is independent of age in itself. I think this is a lot of text. I'm gonna skip this now because I have, I think, more interesting points to make. So we've done up until this point a lot of work trying to understand ecDNA, and for most of that work, we focused on computational methods. I didn't go into all the various things we've done around ecDNA for the sake of time. Suffice to say that we have, as a computational lab, largely relied on high throughput data sets from whole genome sequencing and others to try and understand as best we can the biology of ecDNA. But of course, now that we've discovered that ecDNA exists, and we know that ecDNA is, as I mentioned at the start, it's first and foremost a method for activating oncogenes. It's easy to now make the jump to, okay, well, if you can activate oncogenes via ecDNA, can we now think of ways that we can target ecDNA? And could that be a way to treat cancers? And the reason for ecDNA being an attractive target for anti-cancer therapies is because normal cells don't contain ecDNA, at least not ones of this size and ones that contain oncogenes or genes to begin with. And because we are increasingly uncovering new biological characteristics of ecDNA that appear to not be affecting linear DNAs, such as that ecDNAs can cluster, or that ecDNAs end up in micronuclei, or that ecDNA likely have a specific formation mechanism. So if we can think of ways to block these types of ecDNA specific processes, we actually can block the oncogene that resides on the ecDNA. So in our trajectory as a laboratory, we spent some years in Houston, starting my computational lab there because that way I could stay in the TCGA network. But as a family, my wife and I felt that the Northeast would be better suited to our needs, in part because my wife is actually from Maine. So we wanted to make an effort to be closer to my wife's family. And to make this happen, I relocated the lab to the Jackson Lab. The Jackson Lab is an independent nonprofit research institute. Some of you may know it because Jackson Lab is also a mouse vendor, so you can purchase mice from Jax. But it is a nonprofit, and any surplus income from the mice actually comes back for the research. And around that time, Jax had opened up a second campus in Connecticut, in Farmington, Connecticut, to be precise, and that's where I settled the next phase of my lab. And the good thing about relocating a laboratory is that you have the ability to negotiate. And as we thought that ecDNA may be an anti-cancer therapy target, I felt that we really needed to start doing MATLAB research to further our knowledge on ecDNA and hopefully uncover ways to treat cancer. So we started at JAX in 2016, and this also meant that we started our wet lab operation in 2016. As I mentioned before, ecDNAs are circular and they lack centromeres. We've mapped ecDNAs using long-range sequencing, but our collaborator Paul Michel also showed circularity in the absence of centromeres via, for example, electron microscopy and DEPI staining, as they did in this 2019 paper. We had also concluded on the basis of our computational data sets that ecDNA has to be segregating unevenly, right? That's how we explained the meta-amplification behavior. And consequently, that the presence of ecDNA would significantly increase levels of intratumoral heterogeneity. So as I started at JAX, one of my first orders of business, and I think that's probably true anytime you start or restart a lab, is to find good people to work with. And I feel like I was very fortunate to first hire Amit Gujar, who is still our lab manager, and as well as a senior scientist in the lab, who brilliantly filled in and continues to fill in my lack of knowledge in how to set up a wet lab. So basically, Amit helped me to purchase equipment, to make sure that the consumables were there, make sure that we were sticking to all the regulations surrounding safety and so forth. And at the same time, I was approached by Inhi, who had just graduated from Seoul National University and who was interested in doing a postdoc with us. So Inhi was excited about ecDNA and recognized that we lacked tools to study ecDNA in model systems. So she set out to develop a method to visualize ecDNAs in live cells, to, for example, demonstrate that ecDNAs indeed inherit unevenly. So she designed a CRISPR-based method where we use a DEP-Cas9 in combination with guide RNAs that are loaded essentially with fluorescent molecules. So the guide maps to a ecDNA-specific breakpoint, and because of the fluorescent molecules, we can now visualize ecDNA in live cells. So he conceived and perfected the method, demonstrated that it works. And here's an example of how it works. So here we're looking at one cell that is first pre-mitosis with the arrows indicating the presence of ecDNAs and marked by this signal over there. The cell enters mitosis. At that point, we actually lose our fluorescent signal. And we think this is because the DNA heavily condenses during mitosis. So the guides can no longer access the breakpoints. But here the cell starts to exit mitosis. The fluorescent signal comes back. And at the end, you can appreciate that there's a lot more signal in one cell versus the other, providing indisputable evidence that ecDNA indeed inherits and segregates unevenly. And he repeated this experiment many times. And here we compared daughter cell distributions, basically to show that the distribution of ecDNA molecules is pretty much random. Whereas for linear DNAs, as you might expect, chromosome 7 and HEMOC4 as controls, you see very much a linear correlation between the number of signals in the two offspring cells. Second, and I alluded to this already, and he recognized that in a number of cells, we started to see convergence of ecDNA signals in the same point in the nucleus. So here's one example, a time-lapse series of two ecDNAs that converge over time. And we saw many of such examples. This was sort of a serendipitous finding, if you will. And both ecDNAs come together, and we rarely would see similar, make similar observations for linear areas. Now, this type of clustering behavior could also be a random phenomenon, because in many of these nuclei, there's lots and lots of ecDNAs floating around. So naturally, every now and then, some of them are going to float together. So to determine whether this is a random interaction or not, we compared the length, the duration of ecDNA interactions to the duration of telomere interactions, with the rationale being that there's lots of telomeres in the cell. So if our interactions would be random, we should be able to see similar patterns as for telomeres. So here you can see the duration of various telomere interactions, and as you can see, they're pretty much spread out, and they come and go. Basically, this is what you would think of as a random pattern. But for the ecDNAs, it's clear that these are sustained interactions and less likely to be totally random. And then finally, and he did co-localization studies using immunostaining, for example, to show that, first of all, ecDNA signal tends to co-localize with RNA pol II, being a critical component of the transcriptional machinery. And even more so, and this is just reflective examples, but we actually were able to do statistics to show this, even more so when ecDNAs have clustered, when they have formed ecDNA hubs, the likelihood of RNA pol II co-localization is significantly higher, suggesting that the formation of hubs actually has a role in driving transcription. And to drive that point home, and he did immunostaining of EGFR, and the red signal here is the amount of EGFR present. And to make you appreciate that when there's clusters of ecDNA, these large fluorescent signals there, that you get considerably more EGFR expression, suggesting that hub formation has something to do with driving gene transcription of the ecDNA cargo gene. So I wanted to end on that note to show you what some of the things we are doing in the wet lab. We are now, of course, following up on these observations that ecDNAs form hubs and try to figure out how we can block them. We actually believe we have found additional mechanisms that I haven't disclosed to you yet, through which we think we can effectively target ecDNA. This, in that sense, it's actually a very exciting time in the lab. And in that sense, the recent relocation to Yale, because my lab has been at Yale now for 6 weeks, has actually disturbed our progress a little bit, but we're just about to get back on the, we're just about to reopen the wet lab and restart our studies. So it's very exciting. We think about 10 years ago that we found something that turned out to be quite relevant to glioblastoma evolution. But moreover, it ends up that the mechanism we uncovered 10 years ago through computational analyses may actually end up being a quite relevant target for anti-cancer therapies. And that was also one of the reasons why we started a biotech company a few years ago, Boundless Bio, that I mentioned earlier, to further capitalize on these discoveries from my lab and the labs of several others. So I hope to have also demonstrated to you how dry lab and wet lab can interface effectively. It's really a back and forth in our group. Some studies matriculate from analyses that we've done in silico. For example, we have experiments where we look at radiosensitization, which are directly informed from our in silico work. And vice versa, of course, we are also doing lots of, we are also informing our computational analyses on the basis of discoveries we make in the wet lab. So it's really a back and forth. In the end, to me, they're just tools, right? I don't consider myself a computational biologist. I don't consider myself a wet lab biologist. I consider myself a cancer biologist. I think that's true for everybody in my lab. And computation versus wet lab versus bench versus in vivo work, they're all tools that can be leveraged for different goals. And the same principles of doing science applied, and that you need to have solid controls, that you need to have independent verifications, that you need to have all the toolbox in place and people that know how to use the toolbox. In that sense, I don't really see a difference, and I'm not a big fan of the box thinking of you're a bioinformatician, you're a wet lab scientist. We're all scientists in that sense. And finally, I have been very fortunate to have very good people in the lab for the entire time of the or since opening us and since opening in 2010. Hoon Kim, I still closely collaborate with. He was one of the first 3 recruits. I continue to have very good people in the lab today. Some of them are now starting their own laboratories, which also makes me very proud. And I think this is a common truth, but having good people around you is always the most critical. On that note, I'm very open to recruiting. I just started here, or we just started here, Yale. This is a fun group of people, and that I'm very indebted to for all the at least academic successes we've had so far. I think it's a great environment to be in at Yale University. So if anybody is looking for a next step in their career, they are very welcome to reach out to me in this case. And with that, I'll open the floor for questions and discussions.